Let me remind you that my clock is not quite correct, so I'm going to be at a slightly, what do you call it, slightly delayed schedule because I'm following the clock on my computer and I cannot change the clock on my computer. I got to take it into Clark College to get the time corrected. I know it's probably at least 4 minutes late now. My apologies there, but you're just going to have to be 4 minutes late. Sorry about that. Let's see. So today we're going to talk about, oh, wait a minute, you're not seeing my screen yet. Can somebody confirm that you're hearing me?
Yes.
I can hear you.
If I could also ask you guys to check that I'm actually recording. I'm not sure that the lecture got recorded today.
A notification popped up for both saying that it's being recorded.
Yeah.
Now it's being recorded. My question was the lecture being recorded? And
No, the lecture said it was being recorded as well.
okay, that's good. Thank you. If ever you see it's not recording, please let me know, because once I share my screen, I can't see this note here, which I guess you guys can't see that because I'm not sharing my screen yet. But let me share my screen now. Now I'm showing my screen, and the only way I can tell it's recording is if I look right there and I see that there's a little red dot. I think if I were to blow that up, that might even be an arrow. That's the only way I can tell that the recording is being done. And it's very hard to tell. So today we're going to talk about Lab 1, the ubiquity of microorganisms. You need to also read for next week the infectious disease research project. Oh, good. That's the lecture. That means it was recording. I thought it had finished, and I said, geez, if it finished that soon, it couldn't have recorded. But I don't know. Let's just say I was mistaken, and it obviously was recording, because it took about 13 minutes for the file to complete. I'm actually, that's good. Usually it takes a little longer than that. Anyways, next week you need to read the infectious disease research project. It's a file on Canvas. I'm going to talk about it next week, and I would like you guys to at least look it over, if not read it, so that you can understand the project before I talk about it. Let me close down some of these things I don't need. Oh, don't want to do that. I wonder why Zoom has two things now. Let me go to my class. Oh, there it is. Come here. You go to home or modules, they go to the same spot. So I think it'll be under week 2. All right, let's look under week 1. I'm not seeing the link. Just let me look again. Oh, I'm looking in the wrong place. I should be looking in the lab. That's why I was not finding it. There it is. So click on this and you'll get the project instructions. I'll talk about it, but like I said, at least look it over. It is a lengthy document talking about this project. This will be worth 100 points. It's not due until May 13th. I'm not going to start it until, let's say, Tuesday of next week. So the earliest you can pick a project will be Tuesday of next week. If you pick a project from now until Tuesday, I'll just tell you, hold off, because I'm not allowing you to pick a project yet. Okay? And I'll talk more about that when I talk about the project. Any question about any of that? But I do want you to read the instructions so you know a little bit about the project. If not, let's talk about Lab 01, the ubiquity of microorganisms, and then the worksheet for Lab 1, which I'm not going to use those links. I'm going to use the files that I have on my computer. All right, Lab Module 1, ubiquity of microorganisms. You have a little bit of reading to do before the lab. Read page 157 to 162 on culture media. Read page 162 to 163 on obtaining a pure culture. There are some video clips to watch throughout this lab. Some learning objectives. Know that microorganisms are almost everywhere. Be able to describe the purpose of culture media. Describe what colony abundance is and colony morphology. Define the following terms: inoculation, ubiquity of microorganisms, sterile, isolation, incubation, in vitro. Culture media, we have 3 different types: solid, which can be a auger, and that can be an auger slant or an auger deep, and it can be an auger plate as well. So I guess that'd be 3 different types. And then you can have a liquid broth that we call a broth for our different culture medias. We have different organisms. There's fastidious, or a media which grows fastidious organisms. And then we have non-selective media, and be able to explain what ubiquitous is and a colony. Any questions about any of that? Boy, I don't have this highlighted. Let me see if I missed something. I've only got one link here. All right, I'll have to go back and highlight it. Something happened to my highlighting. Microorganisms are organisms that are too small to be seen by the naked eye. It does include viruses and prions, which are special because they really aren't organisms. They do not have cells, but they do cause disease, and that's why we study them in microbiology. We'll also include helminths, which are worms, parasitic worms. And even though the adults are not microscopic, the infective stages of the helminths usually are microscopic. And of course, some helminths, some worms, cause disease, and that's another reason why microbiologists study them. The phrase ubiquity of microorganism refers to the concept that microorganisms are essentially everywhere. That's not entirely true. You don't find them on lava, but generally you find them everywhere else you look, unless it's a sterile environment. Prokaryotes occupy an amazing diversity of niches on the planet. Some bacteria have been revived after being suspended in amber for tens of millions of years. I think the record is something like 40 million or maybe 400 million years. They had a endospore that had been frozen away in Antarctica, and then they made a drill core of the ice and brought it out and found this endospore and thawed it out and found it started germinating. Microorganisms are in the air we breathe, the food we drink, the water we drink. They're almost everywhere, and this causes some problems because in the lab we generally want to work with a pure culture where there's only one species. And that means that when we do our work, we have to work in an aseptic, use an aseptic technique to continue working with a pure culture. We spend a lot of energy keeping our internal body sterile, free of living microbes. And then if we don't, we get sick when our immune defenses fail. And then we have microbes growing in our blood. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. You're also going to learn basic lab protocols using solid media, labeling lab cultures, procedures for disinfecting the lab bench, and procedures for washing your hands. Generally, when we're growing things in the lab, if you were to come into a lab, I would give you a tube or a plate, and it will be sterile because we spent energy to sterilize it. Usually it'd be sterilized by putting it in the autoclave. A culture medium, also called a growth media, is one that is sterile, and then we can inoculate it with a specific culture of one species to get it to grow. The culture media contains a variety of nutrients that determine the range that the microbes can grow, meaning determines what microbes can grow in it. Culture medias may be liquid. We call these broths, and they're commonly used in a test tube. We can add agar or some other solidifying agent to the liquid broth to make it a solid medium. We can grow the solid media in a petri dish, and we call that a petri plate, or we can grow it in a tube. We have, that's would be a liquid broth, and that would be a deep, an agar deep, where the agar is just flat across. And this is solid media, or we can grow it on a slant. Occasionally you have a deep and a slant, where you have the deep and then a slant going like that. But most of the time it's just a pure slant or a deep. Any questions about any of that? If not, there's a little video you should watch on preparing pour plates. Note how the culture medium needs to be sterilized before using it. Why must culture medias be sterile before we use them? That's a question for you. Anyone know? Anyone guess? All right, that's a question in the lab. You should think about it. We have different types of media on the agar plates. We have general purpose media, and this is the only type of media we're going to talk about. A general purpose media is a non-selective media, meaning we can grow many organisms on it. It doesn't select for any specific type of organism. General purpose media contain complex composition, usually using ground up peptone, ground up yeast, ground up beef extracts, or ground up tryptic soybeans. Okay, so that's a general purpose media. We use them because they're cheap. Many organisms can grow on them. They supply a lot of nutrients, but many organisms cannot grow on them. If the organism requires a lot of additives being added to the media, we call that fastidious. So non-fastidious organisms grow on a general purpose media, like E. coli, Staphylococcus aureus, There are three different types of general purpose media that we'll talk about. The first and the third one, nutrient agar. It's a very simple media that mainly contains ground up beef extract, and I think peptone, which is a protein extracted from soybeans. Nutrient agar with yeast extract is nutrient agar, and we have added to it yeast extract, ground up yeast. And that's a little richer, a little more expensive too than nutrient agar. We also have tryptic soy agar, or TSA, which is another general purpose media. And this one is obtained from soybeans. These are non-selective media. They grow a lot of things, but they would not grow something that requires a specific additive, like Streptococcus pyogenes, which causes strep throat, would not grow on a general purpose media. You need to add specific additives for Streptococcus pyogenes to grow. And what we could add is sheep's red blood cell, and then the red blood cell supplies the nutrients that Streptococcus pyogenes needs to grow. And so it will grow on a blood agar plate, but it will not grow on like tryptic soy agar, which is what blood agar is minus the blood. All you do is take tryptic soy agar and add blood to it, and then you have blood agar. And that would be, like I said, a fastidious organism, one that you need to add specific nutrients to in order for that organism to grow. We do have different types of media. We're not going to talk about them today, but we have selective media, differential media, and reduced media. They will be discussed in a future lab. Whenever you're working with microorganisms in the lab, you should date all cultures, meaning all plates, all test tubes, all other containers, which we don't use in the microbiology lab. You should date everything with your name or your initials. The date, like today, you'd put 4-6 if you were to come into the lab. The type of medium you're using, you can use the abbreviation NAYE for nutrient agar with yeast extract. You should put the inoculum, what you're going to grow on the media. Like this one here is inoculated with the instructor's name, the date, which that looks like 2001, TSA for tryptic soy agar, and then what they're going to grow in here, E. coli. Okay. The temperature that you grow it at is optional. Let's see if he's got that. He's got 37 degrees Celsius is what temperature it was grown at. That's optional. In the lab, we grow things at either 37 degrees, 35 degrees. Let me think about that. Is that 30? I think it's 31 degrees. So 37 degrees, 31 degrees, or room temperature. So an agar plate should be labeled around the perimeter, because if you were to put it here, you wouldn't be able to see what's growing as well on this part of the plate. So you put it on the perimeter of the plate. On a tube, you write it on a piece of tape, and then you put the tape on the test tube. And that's how you properly label a culture, with your name, the date, the type of media, and the inoculum are required. The temperature is optional. So inoculation is when you place cells or microbes into a sterile culture media. There's a number of ways of inoculating, and we'll talk a little bit about that in this lab. Incubation is then placement of the inoculated culture media in an appropriate physical environment for a specific time to allow the growth of microbes. Generally, we incubate our cells in an incubator. And there we're looking at an incubator. This is a fancy one that can control the temperature, what those dials are. It controls the humidity. We put water in this pan there, and that puts water in the air. And this looks like it can control either the carbon dioxide or the oxygen concentration, or both. Most of our incubators only control the temperature and the humidity. If you were to go into our real microbiology lab. All right, any question about that? All right, when we incubate a plate in the incubator, all plates should be incubated. Of course, this isn't going to be very visible to you, so I'll try and make it more colorful. Oh, that's black. Let me get one that's colorful. There we go. So this is the plate. Can you guys see that? Hello?
Yes, we do.
Yeah.
Okay, thank you. You put your bacteria on the plate to make colonies or whatnot. And then when you're done, you flip the plate upside down and then put it in the plate, in the incubator upside down. The reason for doing that is that moisture will come out of the agar and condense on the lid of the plate. And then you'll get droplets of water forming on the lid. And those droplets will fall back onto the agar. And that will disperse the colonies on your plate. It may not give you contamination, okay, because the lid of the plate should be sterile. However, it would increase the risk of contamination, especially if the drip were to come over to the very edge and then come down, because usually the lid is bigger than the bottom. And so this side of the lid would be not sterile, although this side would probably be sterile. And so if you have a drip that comes down off the side, it could contaminate your plate. So it increases the risk of contamination, but the plate should not get contaminated just from the drip of water. The problem is you'll then disperse your growth. And if you're trying to get colonies, you're not going to see colonies. So we put everything in the incubator upside down. That way, if water were to come out of the agar, it will condense on the surface of the agar, and the agar will wick the droplet so that it'll disperse throughout the agar, and it will not drip down on the lid. And even if it were to drip down on the lid, who cares, because we don't grow any bacteria on the lid. Any questions about any of that? All right, I mentioned a colony. That is a bump that we see of cells on the agar surface. Bacteria colonies tend to have different characteristics, but the important reason why we like to grow in colonies is a colony is either formed from one cell or two cells or a clump of cells. So a lot of them are formed from one cell. And if you have a colony from one cell, you know you have a pure culture. because all the colony cells came from one cell. Okay. But when you streak it out, you can get the colony forming from one cell that happens to be streaked in that area of the plate, and there's no other cells near it. Or it could be two cells that are streaked there, or it could be several cells that happen to be streaked there. So you don't really know if the colony is formed from one cell or from several cells. And you don't really know the first time you do the streaking if the colony is pure or not, but about 70% of them will be pure. That's not good enough in microbiology. If you want to say that you have a pure culture, you need to streak from an isolated colony and then streak that out on another plate to get isolated colonies, and then take the cells that have gone through 2 colony isolation events before you can say you're working with a pure culture. And even then, the purity isn't 100%, but the purity will be over 95% if you send the cells through 2 colony isolation events. Okay. Any questions about any of that? All right. A colony is made from at least 1 million cells. If it's below 1 million cells, you can't see it with your eyes, to tens to hundreds of millions of cells. All of them are descendants of one cell or a few cells that got plated in that area of the dish. And what happened if it was plated with one cell is that one cell goes and divides into 2 cells, which go and divide into 4 cells, which go and divide into 8 cells, which go and divide into 16 cells, which go and divide into 32 cells, etc., up until you get at least 1 million cells. And that division by most bacteria is by binary fission, where one parent cell literally splits into 2 identical daughter cells. Here's a little video you can watch on binary fission. And there are some colonies that have been streaked out on a plate. It looks like they went like this, or vice versa, meaning they could have started here. You can notice that the colonies have specific characteristics. There's difference in colors. This one's different than that one. It's different than that one, which is probably different than that one, and certainly different than that one and that one. Okay. They're different in size, different in other characteristics. Like that one's very round. That one has a little funny part to it. That is not round. That's irregular. That's irregular too. You can distinguish the bacterial colonies generally from fungal colonies by the appearance. Bacteria colonies tend to be compact and have a distinct boundary. Fungal colonies tend to be larger. Let me blow this up. The boundary is not distinct. The boundary is hair-like. Even that one is hair-like. And they're also bigger, the fungal colonies. So if you see something like this or like that, it's a pretty good bet it's a fungi. If you see something like that, or like that, it's pretty good bet it's a bacteria. It's not 100% true, but it's greater than 95% true. Let's talk a little bit about colony characteristics. The colonies have different shapes. You can have a circular colony, a rhizoid colony, an irregular colony, a filamentous colony, and a spindle colony. These are the shapes. The margin is the edge of the colony. We call this margin an entire margin. and circular colonies have an entire margin. An undulate colony is mostly irregular, but it could be semicircular, lobate and curled. And these three are so similar, I won't ask you any questions. Is this colony undulate, lobate, or curled? Because that's just too difficult to distinguish. Furthermore, I'd never ask you to distinguish between rhizoid and filamentous, but I may ask you to distinguish between entire, one of these three, or one of these two. Okay. Generally, I would give you this table if I'm asking you a question about the colony characteristics. The elevation, you have to look from the side of the plate. So generally, to see the elevation, you take off the lid and then you look at the side. You can see flat colonies. You can see raised colonies, and those are like a mesa. There's convex colonies and superconvex colonies that we call pulvinate. And then umbinate is the elevations. I would never ask you to distinguish is this colony convex or pulvinate, because I would have a hard time distinguishing those two. The size of the colony is the size of the colony. If it is smaller than the point of a pen, it is punctiform. If it's about the size of the point of the pen, it's a small colony. If it is a large colony, it's the size of the eraser or the cap of the pen to be large or larger. And then anything between the point of the pen and the size of the eraser is a moderate size colony. Any questions about any of that? We can take a look at the colony and determine whether it's smooth or rough. Like that's clearly rough. That is smooth, that's smooth, that's smooth. On a photograph, it's much more difficult to tell if it's smooth or rough. But students, for some reason, have more trouble determining the smoothness or the roughness than anything else. The appearance is whether the colony is shiny or glistening or dull. So that would be a dull colony. Let me blow this up. That would be shiny, that would be shiny, that would be shiny. That's shiny, that's shiny. That's dull. Any question about any of that? Oops, wrong one. All right, there's also the pigmentation of the colony. If the colony has any color, we say the colony is pigmented. So anything that's yellow, red, or what's another color? Orange would be a pigmented colony. So this is a pigmented colony. That's a pigmented colony. This is not a pigmented colony. That is not a pigmented colony. Gray is not a pigmented colony. How about black? Is that pigmented?
No.
Yeah, that's a no. Black is not colored, so that is not pigmented. Usually our colonies are not black, but that's a fungal colony and the inside is black. Ooh. Oh, where was I? Where's the table? Oh, I missed it. Already talked about that. I talked about that. Oh, there's also an optical property, whether the colony is opaque, translucent, or transparent. Most colonies are opaque, a few are translucent, and very few are transparent. Transparent colonies are a little hard to work with because you don't see them very well. I don't see any transparent colonies on this agar dish. A transparent colony would be one you could see through, like you're looking through water. Most of these colonies are opaque, like that's opaque, that's opaque, that's opaque, opaque, opaque. You can't see through an opaque colony at all. Some of the colonies, let me see if I can find one. Here around the edge, meaning that looks like there's either two colonies there or the outer portion is different than the inner portion. The inner portion is opaque, but the outer portion is, the inner portion is opaque. The outer portion is translucent, where you can slightly see through the translucent. So you can think of translucent as lightly muddy water, but you can see through the water. Any questions about any of that? Boy, there aren't very many translucent on that picture. Let me look at another picture. And I don't see any that are translucent here. So we'll go with what few we have here, and that's clearly translucent right there. Oh, there's a little bit of an edge, which is translucent also. I don't see an entire colony, which is translucent. Maybe that one. This would be a round colony. Round colony, very small round colony. That would be an irregular shaped colony. Right there. All right, colony abundance is simply the total number of colonies on an agar plate. Even if two colonies look different from one another, they are probably different species, but you can't be sure without further testing. So colony abundance, all you need to do is count the number of colonies on the plate. That plate has too many. This plate has a lot also, but it's for, let's make that 12, another 30. So let's say this has about 40 colonies on the plate. You have to actually count to get your colony abundance, and then just give the total number of counts. And like I said, this will be about 40, and that's your colony abundance. Any questions about colony abundance?
Does the fungus count in the abundance number?
Yeah, that is if you're talking about microbes, the fungus does count. Doesn't even give opaque because almost all of them are opaque here. The diversity is the number of different kinds of colonies on an agar plate. You use the colony characteristics to identify different colony types. So over here, these three are probably the same. And that's probably true of these, that, this, and that. So there's a large number of, I don't know, orangish colonies on the plate. That one is probably different, so that's 2. That one's different, so that's 3. That one's different, so that's 4. That one's slightly different, so that's 5. That one's different because it's white, although that could be the same as that. So I'll take that back. 4, that's a different colony. 5, that's a different colony. 6. So this one has a diversity of about 7. Okay, now when you give me an abundance number and a diversity number, you don't have to be exact as long as you're close. So if you say something like 5 to 9 here, I'll probably allow it. But if you were to say 20, I would say, no, there's no way there's 20 different colonies on this plate. And then again, if you say 2, I'll also say, no, there's much more than 2 here. Okay. So that's how you do colony abundance and colony diversity. For abundance, you just count the total number. And in this plate, it will be hard counting them right here because the colonies are growing together. Just do your best if they're like that. It'd be very hard to count exactly how many colonies are right here, here, and there. Everywhere else, though, you can count. Like there's 1, 2, 3, 4 right there. But here, I'm not even sure how many are right there. And then that's 2 right there. And this is at least 3. Oops. Well, that's really blown up. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So this one's at least 9. Not in diversity, but in abundance. All right, any questions about any of that? I got lost again. Let me shrink that down. Oh, here we are. Here's a real world example. What is the abundance of cats in this picture? Nobody's going to guess. What's the diversity of cats in this picture?
Abundance would be 7 and diversity would be 2.
Yes, there are 2 different cats, the cheetahs and the lions. There's a male lion, a female lion. So the diversity is 2. And then 1, 2, 3, 4, 5, 6, 7. The abundance is 7. Okay. All right, for this lab, know the terms. And there we have a unique picture, which we'll do something like that today. Or you're going to pretend to because you're doing it online. All right, what is that? Oh, just the answers. Let's see. Let's go to the worksheet. So you're not actually going to be doing this lab, but you do need to understand the principles and what we do. In lab number 1, you're looking at the ubiquity of microorganisms. A number of different environments were sampled by inoculating general purpose culture media with the following. 1 is air, 2 the lab bench, 3 your hands, and you've got hands both before hand washing and after hand washing. Sampling your lips and your tongue, and then sampling the soil. What is in the air you breathe? That's not a question you need to answer. That's just the question of starting procedure 1. Below is a nutrient agar broth plate, nutrient agar plate that was exposed to air and then incubated. After incubation, answer A, what is the colony abundance on this plate? Just do a count. And then what is the colony diversity on the plate? These 2 are probably the same. This one and that one are probably the same. This one looks different. Both of these look different. That one looks different. Those probably the same. So 1, 2, 3, 4, maybe 5 of the small one. 6, 7, 8. So 9, let's count that one too. So there's at least 9 for the colony diversity. Count the number of abundance and put it in A. What step would you take to reduce airborne contamination of a sterile agar plate that you are inoculating? So don't tell me what steps you would take beforehand, and don't tell me what steps you do afterwards. You have to tell me what steps you do while you are inoculating. And there's actually 2 things I can think of. Procedure 2, what gets on your arms when you put your arms on the bench? Once again, this is just asking a question. You don't need to answer this. Go through the procedure of procedure 2. And what we're doing is taking a look at the table before we disinfectant and then after we disinfectant. So that's the picture before, showing you bacteria growing. And there's the picture after, showing you few, if any, bacteria growing. And I can see at least 2. There's one right there. I don't see the other one anymore. Once I blew it up. Did table disinfection work? So answer that for procedure 2A. And then 3, what is the purpose of allowing the table disinfectant to air dry? This one you have to think a little bit about, because I don't think the answer is directly stated in the lab, although the lab module does imply it. Procedure 3, what's on your hands? So you're going to label 4 TSA plates. 2 of them are going to be the hand before, and 2 are going to be the hand after. So you touch your hand to the plates before, right and left hand, then go wash your hands, and we're just pretending, and then touch the plate with your clean hands. They do have to be dried, because if you don't dry them, the water will wick the bacteria from your skin onto the agar plate. And so you want the plate A and plate B to have similar humidity in them. Otherwise, you'd expect more bacteria in the plate, which is decomposing something. Any questions about any of that? All right, so on the left, we have the plates before washing our hands, and on the right, we have the plates after washing our hands. Answer for, is this procedure 3 or 4? Procedure 3, answer, does hand washing work? And that should be a yes or a no with maybe a little explanation. And then 3, suggest one reason why hand washing did not result in the complete removal of microbes. So if we look at the plates after, you'll note that they do have some bacteria growing on them. And why is that the case? Since you washed your hands before you touch the plate in the after plate. And that'll help you with B. Oops, am I in the wrong place again? Yep. This will help you with that. Procedure 4, when you kiss your significant other, what exactly are you giving him or her? So label 2 plates, label 1 lip and 1 tongue. We're giving you the results here. The bacteria from both the lips was grown at 30 degrees, the bacteria from the tongue were grown at 37 degrees, and they were grown for 48 to 72 hours. And then we take a look at the growth. You'll note that there are many colonies in this region, and really this region here too. And it may not be possible to count all of the colonies here. Just do your best if you're asked. I don't think you're asked in this one. And you can see different varieties. And then over here, oops, didn't mean to move the picture. Over here with the tongue, you can see more bacteria. Let me blow that up really, really big. And right there, you can see that you have colonies growing all over here, almost confluently. They're confluent everywhere except for right there. Well, almost everywhere except for right there. You can actually see individual cells right there. That took too long to switch. All right, let's go there. All right, any questions about the tongue and the lips? So which plate has higher colony abundance, the lips or the tongue? And why do you think this is the case, even though you might expect the abundance of both body sites to be the same? That is, what might cause the observed differences in colony abundance? Why would one have more cells than the other? You will have to think a little bit about this answer. It won't directly be answered in the lab module, but most students will get it after they've thought about it for a little bit, for a while. And then C, answer, why was the tongue plate incubated at a higher temperature than the lip plates? Procedure 5, are there microorganisms in the soil? We actually take diluted soil, put it into a tube, then use that tube to inoculate the surface of this agar plate, and then answer the questions. What's the colony abundance? Is it even possible to accurately answer this question? And please explain your answer. You do need to explain your answer because it depends how you're answering, whether this answer is yes or no. Meaning it depends on your further answer, whether yes or no is correct. And then is there greater abundance of colonies from the soil compared to the lips? Compare this plate to the lip plate right here. All right, are there any questions? If not, I'm going to turn it over to you and I will be here until 8 o'clock to answer any questions. Please remember that if you get together with other students, you cannot work on the lab worksheet and then turn that in for extra credit. Actually, this is week 1, so you can't turn in extra credit, but in week 2, you will be able to turn in extra credit worksheet, getting together and studying with someone, but you cannot work on the lab worksheet while you're doing the extra credit. I'm giving you points for the lab worksheet, so get your points from doing it as a lab project. Any questions about any of that? All right, start working in the lab, and I'm gonna stop recording and stop sharing.